Hello students, welcome back to the next session of chapter 16, Heredity and Variation. Today we will study about RNA, that is ribonucleic acid, and we will also look into Mendel's experiments on heredity. Now RNA is the second important nucleic acid of the cell. This nucleic acid is made up of ribose sugar, phosphate molecules and four types of nitrogenous bases: adenine, guanine, cytosine and uracil. The nucleotide, that is smallest unit of the chain of the RNA molecule, is formed by a combination of a ribose sugar, phosphate molecule and one of the nitrogenous bases. Large number of nucleotides are bonded together to form the macromolecule of RNA. Now let us come to the types of RNA. Depending upon their functions, RNA can be classified into the following categories. First, we have the ribosomal RNA, or it is also known as rRNA. The molecule of RNA, which is a component of the ribosome organelle, is called a ribosomal RNA. Ribosomes perform the function of protein synthesis. The second type is messenger RNA, or it is also known as mRNA. RNA molecule that carries the information of protein synthesis from the genes, that is DNA chain in the cell nucleus, to ribosomes in the cytoplasm which produce the proteins, is called messenger RNA. Then comes transfer RNA or tRNA. The RNA molecule which, according to the message of the mRNA, carries the amino acid up to the ribosomes, that is termed as transfer RNA or tRNA. Now we come to Mendel's principles of heredity. Now genetic material is transferred in equal quantity from the parents to the progeny. Principles of heredity are based on this fact: if both the parents make equal contribution to the inheritance of characteristics, then which characteristics will appear in the progeny? Mendel carried out research in this direction and put forth the principles of heredity responsible for such inheritance. The experiments performed by Mendel almost a century ago are quite astonishing. All of Mendel's experiments were based upon the visible characteristics of the pea plant, Pisum sativum, and these characteristics chosen by Mendel were as follows. So this is Gregor Johann Mendel. So let us see the 7 mutually contrasting visible characteristics of the pea plant. First is the shape of the seed. Round is the dominant characteristic and wrinkled is the recessive characteristic. Color of the seed, yellow, dominant characteristic, and green, recessive characteristic. Color of the flower, purple, dominant, white, recessive. Shape of the pod, inflated, dominant characteristic, and constricted, recessive characteristic. Color of the pod, green, dominant characteristic, yellow, recessive characteristic. Position of the flower, axillary, dominant characteristic, and terminal. Terminal means at the tip of the plant, and axillary means in the axil of the stem. Okay. height of the plant, tall, dominant characteristic, and dwarf, recessive characteristic. Now some dominant and recessive characteristics of human beings. Rolling tongue is a dominant characteristic, while its recessive characteristic is non-rolling. Presence of hair on the arms, absence of hair on the arms. Black and curly hair, brown and straight hair. Free earlobe, attached earlobe. So these are some of the dominant and recessive characters of human beings. Now we'll come first to the monohybrid cross. Now in this experiment, Mendel brought about the cross between two pea plants with only one pair of contrasting characters, and this is the reason why it is called a monohybrid cross. So as to study the monohybrid ratio, let us consider the characteristic plant height, only one characteristic, with a pair of contrasting characteristics, that is tall plant and dwarf plant. Now we come first to the parental generation P1. Tall pea plants and dwarf pea plants were used in this cross, and that is why this is called the parental generation P1. Mendel referred to the tall and dwarf plants as dominant and recessive respectively. The tall plant was referred to as dominant because all the plants in the next generation were tall, and the dwarf plant was referred to as recessive because this characteristic did not appear in the next generation at all. That is, it was masked by the dominant characteristic. Now this experiment has been presented by the Punnett square method as shown. Depending upon these observations, Mendel proposed that the factors responsible for inheritance of characteristics, they are present in pairs, and today we refer to those factors as genes. Dominant genes are always denoted by capital letters, while the recessive ones are denoted by small letters. As the genes are present in pairs, so tall plants will be denoted by capital T, that is dominant characteristic, and dwarf plants are denoted by small t. So that is the recessive characteristic. Now during gametogenesis, these genes separate from each other, and due to this, two types of gametes, one containing the factor for the tall gene and the other containing the factor for the short gene, are formed. So you can see here Mendel's experiment of the monohybrid cross, parental generation P1. Phenotypes are tall and dwarf. Genotype, you can see capital T in pair representing the tall characteristic and small t representing the dwarf characteristic. So the gamete pairs formed will be capital T and small t. Now in the first filial generation, you can see that the plant obtained is a tall one, okay, due to fusion of the gametes. Now after selfing in the first filial generation, you can see in the Punnett square, the gametes have been arranged. The genotype is capital T, small t, okay, and the gametes are capital T and small t for both the parents, okay. Now the male gametes have been arranged and female gametes, okay. Male gamete is in the row and female gametes in the column. Now if you pair, you can see the progeny obtained. First pair is both capital T representing pure tall. Then you have capital T and small t, which will also be tall because it has got the dominant gene of tallness. Then comes small t, capital T. Again here also the dominant gene is of the tall one. And finally you have small t, that is both are the recessive genes. So this plant will be a dwarf one. Now here we can see the phenotypic ratio. Phenotype denotes the external appearance of the plant, while the genotype tells you its actual genetic constitution. So phenotype ratio for monohybrid cross is 3 tall is to 1 dwarf. That means 75% of the plants obtained in the next generation were tall, whereas 25% of the plants obtained were dwarf. And the genotypic ratio in the F2 generation: pure tall 1 plant, hybrid tall 2 plants, and pure dwarf 1 plant. So you can see the ratio, genotypic ratio is 1 is to 2 is to 1. Okay, whereas the phenotypic ratio is 3 is to 1. Is that clear, children? You need to know the Punnett square combination of the genes as well as the phenotype and genotype for both monohybrid as well as the dihybrid cross.